In the next few lectures, we will focus on the applications of constraint based modeling. So, in today's lecture, I will show you how we went about identifying drug targets for tuberculosis by performing a flux balance analysis of the mycolic acid pathway in Mycobacterium tuberculosis. Welcome back. Let us study some applications of constraint based analysis today. So, there are many applications of constraint based analysis, but the most popular ones, the practical applications, lie in the area of metabolic engineering and drug target identification, and we look at both examples today, some of them from my own work and some of them from other interesting studies that have been published. So, what can genome scale metabolic networks tell us? This is a nice picture which tells you the distribution of studies that are based only on the E. coli reconstruction. There is a popular reconstruction for E. coli known as the IAF 1260. It accounts for 1260 ORFs, open reading frames, in E. coli. It was published in I think 2012 or so. So, there are about 248 studies that use the E. coli reconstruction, probably earlier than 2012. So, there are about 248 studies that use the E. coli reconstruction, and if you see, about 29% of them study biological network properties, what reactions are coupled, what reactions are interacting with one another and so on, and then about 1/4 of the studies focus on predicting cellular phenotypes, and there is another bunch of studies that do model driven discovery of what are the what is the function of a particular gene or things like that, and metabolic engineering studies again very popular and so on, right. So, a lot of studies work on metabolic engineering and on analysis of biological network properties, and maybe some phenotypic analysis trying to delete prediction of cellular phenotypes, right, and you predict cellular phenotypes again for metabolic engineering or for drug target identification. So, two classic applications. So, let us see a few examples. So, let us see how we identify drug targets for tuberculosis. So, how would you start any of these modeling exercises? You need to essentially figure out what is the goal of the modeling, right. So, if I am trying to target tuberculosis, the obvious idea would be to try and basically knock out the TB metabolism, right, or identify proteins that when inhibited or those are drug targets which when knocked off will stop TB from growing or will actually kill it, right. So, how do you identify such targets? That becomes the first challenge, and I will show you two approaches, a simple approach that is just based on a single metabolic pathway and a more comprehensive approach that takes into account as much as possible information from the whole genome. So, the mycobacterial cell wall is very distinctive and it is associated with pathogenicity, and it contains arabinogalactan mycolate, which is covalently linked with peptidoglycan, which is a more common cell wall component, and trehalose dimycolate, which provides a thick layer. So, this thick layer helps it evade classic antibiotics and also the host immune system. So, you can't take like a regular penicillin or any of those drugs to combat TB. Right. And mycolic acids are critical components of the cell wall. They are basically long chain alpha alkyl beta hydroxy fatty acids. So, there are two interesting fatty acid cycles, I will show them in a moment, and it is also the known target for drugs like isoniazid and ethionamide. These are two very popular drugs, anti-TB drugs. So, this is how the pathway looks like. So, if you just zoom in a little, so there are 4 different sections here. So, the first is basically production of some precursor molecules, then is what is known as the phase 1 cycle, and then what is known as the phase 2 cycle, followed by the production of the final production of mycolates. There are 5 different types of mycolates, we will get to that in a moment. There are 4 main sections. So, this just involves malonyl CoA production, and this is a cyclic set of reactions. So, you see that there are about 21 reactions here from reaction number 2 to 22, which are catalyzed by the fatty acid synthase 1 enzyme. It is a very interesting enzyme. It has about 3000 amino acids and 5 different subunits that basically do 5 interesting catalytic activities. Here you have the phase 2 cycle where you have 5 different enzymes which does the same job, right, and phase 1 operates till a length of C24 and phase 2 starts at around 24 and goes all the way up till 70, 80 or so, right. So, till about 50 or 60, right. So, there are such large chains of fatty acids that are formed, and then finally there are some desaturations, there are some desaturases that act and create some double bonds and things like that. So, you have something known as alpha mycolate, cis methoxymycolate, cis keto mycolate, and trans methoxymycolate and trans keto mycolate. So, there are 5 different mycolic acids that are present in the in Mycobacterium tuberculosis. So, and if you see there is a reaction here which involves the protein INHA, which is the target for known drugs such as isoniazid and ethionamide. So, this pathway is already the target for known TB drugs. So, what we did was, the first obvious task is to go and reconstruct the pathway, which means go through a lot of literature and databases and figure out what are all the reactions. We identified about 219 reactions in just this pathway alone, with about 28 proteins, 28 enzymes, 197 metabolites and 28 exchange fluxes. So, what is the size of the stoichiometric matrix? Right, it is going to be 197 cross 219, but you have to add the exchange fluxes as well, which will make it 247. So, 197 cross 219 plus 28, which is 247, and the model was primarily constructed from cake biopsych and literature, simulated using FBA and MoMA. What is the objective function for a small model such as this? It is a little trickier, right. For a normal cell you would say biomass, right, whereas if I am just modeling a pathway, how would I come up with an objective function, right. So, you need to figure out what is the goal of this pathway, or what is the constraint on this pathway essentially. So, we hypothesized that we would try to produce the most important mycolates, right. So, there are 5 mycolates. So, there is alpha mycolate, cis methoxy, cis keto, trans methoxy, and trans keto. So, one way to do this would be, you could combine all this in some A is to B to T is to C D E ratio and produce something known as mycolate biomass, right, and then you maximize this. The problem with this is, it obviously requires all 5 to be present in a particular proportion, where it is known that the cell does not produce all 5 simultaneously. I mean, it can produce, but there are cases when if it cannot produce this, it will produce more of this. So, then you can't use this approach. So, this is like a classic biomass, right. So, you say A plus B plus C gives some X biomass. If I were to maximize this, if even one of these is not there, this reaction goes off and my objective function will drop to 0. But it is known that Mycobacterium can grow in the absence of alpha mycolate, although a little slowly, right. So, then this does not become a valid objective function. So, we said the objective function should actually just be this. It is some sort of prioritizing the mycolates in the order in which they are important, right. Alpha is the most important, which means that A is much greater than B and so on, but then B, C, D are all like relatively similar orders of magnitude and so on. So, we set up an objective function of that sort and went ahead and did an FBA with multiple conditions. So, first is wild type, so normal Mycobacterium tuberculosis, and then we looked at deleting multiple genes or inhibiting a particular gene and so on. So, let us look at this first.
It's a hypothetical function.
Yeah. So, this is a hypothetical reaction, right. So, we discussed this before as well, this is important, right. If you have a reaction of this sort, if no B is produced, no X is produced, because this is like a hypothetical reaction now. This is even though fictitious, it is a reaction in the model right now. So, if you remember, we had some some X moles of glucose plus Y moles of alanine plus Z moles of ATP gives some K moles of biomass, right. So, if any of these is not present in the particular proportion, or it is not present at all, if something is not present in the particular proportion, it is okay. So, it will be limited by the the one that is at the lowest rate, but if there is no B at all, this reaction can't even happen, right. So, if you know that this reaction can happen, or the cell can survive, or it can produce biomass. In the absence of these, then you have to use a different objective function. Right? So finally, you are trying to make an objective function that will try to best capture the lethals and so on. Now if I do a single gene deletion and so on, how does it agree with what's reported in literature? That will become my target. So let's see how that happened. So this is without gene deletions. So we are just plotting the flux distribution here. So and on a relative scale, right? So -1 to +1. You can think of this as -1000 to +1000, whatever bounds that you give. And you see that some flux is very high, nearly 1, and another flux, I think this is for acetyl and malonyl CoA. And then the cycle, all the reactions actually have a very similar flux, right? And finally, this part is where you have the mycolates, right? So you have alpha mycolate is reaction number 197, which has some flux of 0.029 or something like that, and these are the other mycolates, right? So you have all the mycolates being listed here, and you see here that there is a redistribution. When I delete PCAA, which is an important gene for alpha mycolate production, you see that there is a redistribution of the fluxes here. And when I delete INHA, it all goes to 0 basically. There are only some exchange fluxes which are not even relevant. And here under the inhibition of mycolate INHA, you again see that most fluxes drop to 0. Right? You look at the scale, it's all very low. So this scale is very different from what you have in the previous plot. So and how do you simulate inhibition? We basically fix the UB to 10% of the value. If you have 90% inhibition, you fix all fix the UB of this reaction to 10% of its wild type value. So that's how we studied inhibition. So as I said, we used an objective function prioritizing the mycolic acids based on the cell wall composition of mycolic acids, and we identified 16 genes as essential, which had good agreement with experimentally available gene essentiality data. So they are experimentally determined data using a technique called transposon site hybridization mutagenesis. So what it does is it knocks out every single gene in Mycobacterium and reports the phenotype. And essentially we had 19 agreements with this data, about 5 disagreements, and for 4 the results were not available. And it turns out though that in the Sassetti paper, INHA itself is not reported as an essential gene, even though it is a very well known drug target. And but once you have a short list of targets, there are other factors to consider, right. So what would be a good drug target? It should not have any similarity with human proteins, right, because you do not want a drug that you are targeting against tuberculosis to go and damage any human or like bind to any human protein. It could cause an unwanted side effect, what we call as adverse drug reactions. So to eliminate these, we just did a simple sequence analysis. So we looked for human proteins that had a homology of more than 30-40% with tuberculosis and eliminated those, right. So that helps you to make sure that these targets are going to be less likely to bind to human proteins. You can do better than this, as I will discuss a little later. So this is just a very simplistic approach, just doing sequence analysis, but you could do structure, or there are other things you can do. We will just come back to it with the next example. Today's video, I introduced you to the different applications of flux balance analysis and constraint based modeling, and I hope the example we gave was quite motivating, which was one of drug target identification for tuberculosis, wherein we looked at the all important mycolic acid pathway in Mycobacterium tuberculosis to identify critical reactions or enzymes which could potentially be targeted. In the next video, we will switch gears and look at another kind of application, which is essentially metabolic engineering, and we look at how a very interesting approach was used to identify targets for improving lycopene biosynthesis in E. coli.